Thank you. Once again, just to extend our welcome to everybody who's joined us today on the webinar. This presentation is going to cover the importance of enteritis control in laying hens. The economic impact of enteritis is most quantifiable in broilers. This is where the rapid life cycles allow for precise calculations of the body weight gain and feed conversion rate. In layers, however, where fine measurement of feed conversion is difficult to achieve, the cost of mild enteritis often goes unnoticed or remains hidden in the flock. But the impact is there, and it's estimated to be costing up to 6 eggs per lifetime of the layer. This presentation is going to look at the importance of intestinal health, the effects of enteritis, implications of enteritis, and how to diagnose the problem in your flock. A healthy gut is essential for digestion or breakdown of the feed into its basic components such as proteins, sugars, and minerals. These nutrients are then absorbed across the intestinal barrier, where they enter the bloodstream and are made available to the bird for growth and other physiological functions. It is not only nutrients that are found in the lumen of the poultry gut, but also a large resident population of microorganisms. It is estimated that there are more than 10 to the power of 12 organisms per gram of intestinal content. This microflora population contains beneficial bacteria that are essential for digestion of the feed components. But not all the organisms are beneficial. Some can be harmful or even toxin producing. To protect the bird from potential pathogens, the gut has developed a thick intestinal mucosal barrier. Activated B lymphocytes interact with absorptive cells, immune cells, and the microbiota in the gut to produce signals that cause goblet cells to produce a thick layer of mucin. This then forms the mucus barrier that provides a physical barrier between the epithelial cells and the potential pathogens. B lymphocytes in the gut-associated lymphoid tissue, like Peyer's patches, together with intraepithelial plasmatic cells, produce high levels of secretory immunoglobulin A and immunoglobulin M antibodies, which enter the mucus layer and inhibit surface colonization of microorganisms, thereby dampening the penetration of potential antigens. The list of potential pathogens found in the gut is extensive. They include viruses, such as reovirus, pathogenic bacteria like Clostridia and E. coli, protozoa like Eimeria, which causes coccidiosis. Toxins like mycotoxins or rancid fatty acids are also known to damage the gut, and of course infection with Ascaris intestinal parasites. Now, pathogens have a direct effect on the gut, causing enteritis, but we often forget about stress events that result in reduced feed intake. The resulting reduction of feed and reduced movement in the gut allows for a buildup of potential pathogens. Also, any factor that causes an increased stress can suppress the bird's immune system and offset the balance of microflora in the gastrointestinal system. Necrotic enteritis, as a result of feed denial in broiler breeders, is a common occurrence. Layer birds or pullets could have reduced feed intake over the change of a diet, for say, going from a starter crumb to a grower mash, or even result from a change in raw materials. The transfer from the grower house to the layer house is a stressful time as the bird needs to learn the new system of feeders and drinkers. In addition, it is at this time that the reproductive system is developing. The physiological changes require additional nutrients and energy, which the feed intake must provide. As she comes into production, she is laying an egg a day, which is increasing in size. Not only that, but the layer is still growing. So now let's look at the effect of enteritis on the intestine. Under normal conditions, food passes through the intestine and some of the nutrients are digested by epithelial cells in the intestinal wall. Pathogens cause damage to the intestinal villi and the mucosal barrier is disrupted. Inflammation of the intestine will lead to increasing numbers of lymphocytes and other immune cells within the intestinal mucosa. These white cells are loaded with antimicrobial molecules. However, most of these molecules also have the ability to cause tissue damage. Tissue that has been damaged in response to the immune reaction will release pro-inflammatory signals, leading to higher levels of inflammation. Poor nutrient absorption and undigested feed is now a substrate for clostridial growth. In this way, enteritis can become a self-perpetuating disease. The implications of enteritis are that after only 12 hours of inflammation, a bird can lose up to from 20 to 35% of the absorptive gut villi surface. The villi are these finger-like projections that line the intestine and facilitate nutrient absorption. Enteritis impacts villi growth, causing a reduction in the length of the villi, leading to a reduction in the absorptive capacity. Although villi can regrow, dependent upon the extent of the initial damage, the reduction in villi length will have already affected the feed conversion rate, thereby limiting the body weight gain or the egg size. Not all animals within a flock respond equally to a similar challenge. Some animals will be more affected, some may die, and some will be almost unaffected. This phenomenon is typical of a large population, and in layers, it has an impact on the flock's uniformity. It is well accepted that bringing flocks into lay without optimum body weight limits the lifetime performance. Flocks cannot be managed by exception. The average condition of the birds will dictate the flock management as a whole. Nutrition and management of environmental factors will reflect the needs of the majority of the population. The more animals that deviate from the average, the less we are able to meet the requirements for optimal production. So we end up compensating in flocks that show enteritis. We have to hold them back. Holding them back means more time in the grower house and more cost to the pullet. Flocks with enteritis coming into lay will be difficult to manage, as the affected individuals are in different physiological stages of egg production. The result is the flocks are late into lay. They do not peak, or they peak very late. They do not achieve their target egg weight, and ultimately the result is an economic loss. Looking for signs of enteritis should be the task of every poultry worker on a day-to-day basis. Each worker should be familiar with the clinical presentation of the disease, note the flock behavior, and be on the lookout for signs of diarrhea. Ideally, the clinical signs of enteritis will be picked up by the house worker before production drops are seen and the flock starts losing body weight and/or egg weight. In order to be able to spot diarrhea or abnormal feces, we first need to be able to identify what is normal. Normal feces is brown or greenish in color with a white spot on top. Since birds do not urinate like other animals, the white spot on top of the feces is a product of the urinary system. So the spot is made up of urates, combined with waste that is being passed through the intestine. Broody hens, who nest continuously throughout the day, will have another type of dropping. They do not want to relieve themselves in the nest where the eggs are, so they hold on to it for a long period of time. This usually amounts to them relieving themselves only once or twice a day. As a result, the feces of a broody hen will be much larger than normal, sometimes 3 to 4 times as large, and with much more of a potent smell to it. This is in fact a normal feces, but one that is originated from the ceca. The ceca is a branch of intestine that absorbs water from the feces to aid in fermentation of the food that has not previously been broken down enough. This area must be eliminated or emptied a few times a day, and results in feces with a very strong odor as well as different colors and textures. It is often gooey in consistency and ranges in color from black to yellow. This may look abnormal, but is in fact normal for birds that have got access to the range. Here the bird has simply eaten a lot of plant material, giving the green discoloration to the feces. So here are a few examples of abnormal feces. Sometimes diarrhea is in fact caused by disease. See if I can get this highlighted. There we go. The top left corner is an example of pylorem disease or bacillary white diarrhea caused by Salmonella pylorum. These two pictures in the middle here are an example of a worm burden or perhaps early onset necrotic enteritis. Clinical necrotic enteritis will look like these pictures over here, the top right and the bottom left hand corner. And the green diarrhea in the bottom here has been associated with diseases like Newcastle disease and even Gumboro disease. The green stain is from undigested bile and rapid passage of the intestinal contents through the gut. Most often when blood is observed, it is due to coccidiosis. The redder the blood seen, the more caudal the infection in the intestine, the less time that the blood has had to be digested, and the fresher that it appears. When dark feces is seen, this is an indication of bleeding into the upper intestine. The red blood cells have been broken down by digestion, giving the feces the darker color. Worm burdens can also lead to enteritis and diarrhea. Roundworms, seen here on the left, are a problem and need to be treated. Tapeworm, seen in the feces on the right, does not seem to cause enteritis directly, but nonetheless needs to be treated from a welfare and a personnel point of view. There is little to no value in evaluating the gut of an old mortality. The abundance of bacteria in the lumen leads to rapid changes in the gut and the surrounding tissues after death. For this reason, when evaluating gut health, fresh samples are needed. The photos here were from an outbreak of necrotic enteritis. On the left, bloating and abnormal coloration of the gut on the external surface is observed. When opening the intestine, hemorrhagic necrotic material is visible within the lumen. When that material is removed on the right, the damage is visible to the intestinal lining of the gut. Here is an example of severe focal duodenal necrosis. The lesions are observed in the duodenal loop. This type of infection can result in a 10% production drop along with an egg weight reduction of about 2.5 grams per egg. So how to proceed when we identify enteritis in a flock? Any intervention should be in consultation with your veterinarian first. The diagnosis of the underlying cause will require the expertise of your veterinarian and possibly further laboratory workup. The treatment needs to be specific for the problem, otherwise we are simply wasting money, and in the long term we are prolonging the recovery of the flock. There are a number of products on the market now following the ban of antibiotic growth promoters for gut health. These products are made up of plant-based oils or derivatives that promote gut health and function. These products seem to have little treatment benefit, but there seems to be value in using them for preventative or prophylactic approaches. Some of these plant additives can be classified as prebiotics. These are plant fibers that act as food for the good bacteria in the gut and stimulate growth of the pre-existing good microflora. Probiotics are the actual living strains of bacteria that add to the population of the good microflora. These products can be made up of single bacteria species or a host of harvested bacteria from a healthy gut. There are chemical anti-inflammatory options, but these really would be only used as a treatment option. Feed acidification is becoming more popular. Butyric acid has been shown to improve the tight junctions of intestinal epithelium, thereby strengthening the intestinal barrier and the upregulation of tight junction proteins. In conclusion, enteritis in a flock will result in economic losses in terms of potential feed conversion, mortalities, production, and of course the treatment cost. There is also an unseen localization of the immune cells to the site of inflammation, which leaves the bird potentially exposed to other pathogens. The good news is that enteritis is entirely preventable. Attention to flock health monitoring will hopefully bring on early intervention should the problem arise and reduce the impact on the flock. Again, just thank you for your time. For additional questions or information, please don't hesitate to contact your local Highline representative.